أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم يا الله يا رحمن يا رحيم يا مقلب القلوب ثبت قلبي على دينك. Today we are going to discuss little bit about my research work done in 2007. My topic of research was preparation and optimization of ready-to-use kit conditions for swab test on animal food, which is abbreviated as STAF, STAF. So before discussing all the details, I would like to share a little bit about the introduction of this research work. Major sources of animal proteins, all of you know that they are meat, milk and eggs, which contain all the essential amino acids which are required for normal growth of the human beings. And total requirement of animal proteins, they are increasing with increasing population of humans, and proper care of quality and health safety of animal protein sources has become basic need of the world. So today, basic need of the world is the quality, proper care of quality and health safety of animal protein sources. During last decade, irrational use of antibiotics in animals and poultry has been noticed. In order to save them from infectious diseases and to improve their production in the form of meat, milk and eggs. And this careless and irrational use of antibiotics in livestock as well as in poultry may lead towards antibiotic resistance problem in human beings. Therefore, great effort has been directed towards the proper management and monitoring of antibiotic residues in meat, milk and eggs. So this introduction is providing the base for my research work. Antibiotics in milk may lead to allergic problems in consumers, and also they may cause losses in fermented food production because of inhibitory effects on culturing process. And this was noted in 2003. It is necessary to monitor the presence of antibiotic residues in the animals before slaughtering, so that the consumers may be avoided, may be prevented from these allergic reactions and from this resistance problem. Pre-harvesting antibiotic screening test, which is abbreviated as FAST, P-H-A-S-T, FAST, and in-plant screen test. This was developed in 1989 to improve the capability of the antibiotic detection program. This FAST test was developed in 1989, and the purpose of developing this test was to improve the capability of antibiotic detection program. So this test may detect a wide range of antibiotics in 1998. To check antibiotic residues in red meat species, swab test on premises was used. Swab test on premises, abbreviated as STOP, STOP. Recently, two main names Sajjad and Jabbar in 2005 have modified these two above mentioned tests, FAST and STAF, to check antibiotic residues in meat, milk and eggs. So in this context, the present study, my research work was conducted to develop, to prepare and to optimize, to standardize a ready-to-use kit conditions for swab test on animal food for the detection of antibiotic residues in animal food. And this kit was named as STAF kit. STAF. STAF means swab test on animal food. There were three basic conditions which were optimized in this study. Number one, thickness of media. This was optimized and standardized. Number two, spore count of Bacillus subtilis. And number three, incubation temperature. These three basic conditions were optimized during this study so that a kit may be developed for the detection of antibiotic residues in milk, egg, meat, and all products of animals. Objectives of my study were to get rapid results as early as possible under field conditions. This was the main objective of my study, that number one, to get rapid results as early as possible under field conditions, so that animals having residues, they may be retained and slaughtering may be delayed, so that problem of resistance, problem of allergic conditions, problem of diseases created by the antibiotic residues, they may be stopped. And to determine, another objective, to determine the feasibility of STAF kit under field condition. And also another objective was that also to get more visible zones of inhibition under field conditions. So again, I want to repeat the objectives of my study. Number one, to get rapid results as early as possible under field conditions. Number two, animal slaughtering having residues, they may be retained, animal slaughtering may be delayed, animal slaughtering may be delayed, and to determine the feasibility of STAF kit under field condition. And next objective was to get more visible zones of inhibition under field conditions. So this was little bit about the introduction of my research work, which may provide the base of understanding that why my research work was conducted. In next lecture we will discuss all about the research process, inshallah al aziz. Thank you very much.